In this lecture, as we continue our survey of metazoan development, we have now reached the final evolutionary level of the higher vertebrates, the amniotes, which include reptiles, birds, and mammals. Amniotes are those vertebrates where embryos form an amnion, or a water sac, which allows their embryonic development to take place on land as opposed to requiring an external aquatic environment as the fish and amphibians do. The organisms that are amnions include reptiles, birds, and mammals. With regard to all of these animals, what they have in common is the amniote egg. In other words, the egg that has the water inside. And there's one branch, which is the reptilian and bird branch, and many systematic biologists actually categorize these as the same group of vertebrates. In fact, the bird is considered to be a clan of the reptile, and it certainly is. Other than it has feathers, it's a very similar kind of organism. The mammals, of course, have mammary glands, and there are a couple of different branches there. The monotremes, which are the egg-laying mammals, and the placental mammals, which we are more familiar with here, and the example that we often use in the laboratory is the mouse. What we'll talk about in this lecture will be development in the bird, embryonic and developmental biology of bird development. Before we get to that, I did want to mention some similarities between birds and mammals. Certainly if you come to the level of the embryo, this is a bird egg now, and this is showing the placental area in a developing mammal. This happens to be the mouse in this case. But if you look at the embryo proper, the birds and the mammals are virtually identical in appearance. The only difference is that the way they get their nutrition and the way they are able to survive during that embryonic period. If we look at the bird, of course, there's a shell in the egg, and if it's a reptile, it may be not a calcified shell, but a leathery shell. But in any event, a shell with some membranes. There's a yolk sac that provides the nutrients for the developing embryo, along with oxygenation. You can see there are some blood vessels on here that produce blood cells as well as produce blood vessels that will help to oxygenate the embryo. The same is true with this sac that's referred to as the allantois. Now, there are remnants of both yolk sac and allantois in the mammal, but most of their functions are taken over with the placenta. In the case of the allantois, this collects the waste of metabolism, the nitrogenous wastes, and other harmful materials that might contaminate the embryo were it to come in contact with the embryo. Also, here you can see there are some blood vessels again partially to provide oxygenation, but also to retain some of the water, to reabsorb the water that's been used to flush out some of these noxious materials. Here again, if you compare the embryonic bodies of the bird and the mammal at this early stage, they are virtually identical. Let's now talk briefly about cleavage. This shows, again, a bird egg. We're talking about the chick as an example, but all of the avian species are virtually identical to this. Here you can see the structure of the egg. There's the shell again. There are a couple of membranes, the inner and outer shell membranes. Here's are some protein strands that are called the chalaza. The chalaza is actually a structure that will hold the yolk and the embryo, the blastodisc, in other words, this cell, in position so that it doesn't get smashed against the side of the egg. So it suspends it in this albumin area, which is the white part of the egg, I'm sure you've seen that. And the yolk is where most of the nutrient material is actually in the egg. Fertilization of the chick egg occurs in the hen's oviduct before the albumin and shell are secreted to cover it. Like the egg of the zebrafish, the chick egg is telolecithal, with a small disc of cytoplasm, the blastodisc. And that blastodisc basically is really the developing embryo then. Like fish eggs, the yolky eggs of the birds undergo discoidal meroblastic cleavage. And what that means is that there's so much yolk that cleavage can't go through all the entire cell, keeping in mind that a bird's egg is the largest single cell there is on Earth. Not necessarily a chick, but birds in general have very large eggs, as you can see. Cleavage occurs only in the blastodisc, which is located at the animal pole of the egg. The first cleavage, which we can look at down here diagrammatically, extends across what's called the area pellucida. And this is an area right here, area pellucida, that's going to form the final embryo. There's also an area opaca composed of some cells that will turn out to later become extraembryonic membranes, such as the allantois or the yolk sac. But the embryo proper comes from this area pellucida. And so initially you have a cell division in this direction, and then you have a number of cell divisions in that area pellucida that gives rise to cells that initially are open to the yolk so that they can get nutrients directly, and they're also connected with each other. But as you can see, the embryo is beginning to develop as a disc on top of that yolk. And this single layer of cells here is called the blastoderm, and these cells are called blastodiscs. This one cell thick area of blastodisc is present when the egg is laid. If you look at the egg as it's coming down through the oviduct to be laid, development is actually taking place. And let's look now at gastrulation. This is a diagrammatic representation of gastrulation. This shows it from a longitudinal viewpoint. Here is the anterior part of the embryo, the posterior part of the embryo. This is the epiblast, area opaca and epiblast. These are the hypoblast areas down below, so epiblast and hypoblast. This shows a view of the embryo from a ventral aspect. In other words, if you were down in the yolk looking up at the developing embryo, this is what it looks like, and this is a view of the embryo viewed from the dorsal aspect looking down on the embryo, which is what you would do if you were to look at an embryonic specimen through a port in, let's say, an egg that maybe you were examining. By the time a hen has laid an egg, the blastoderm contains about 50,000 cells, so it's pretty significant. And a lot of these cells now then have started to form into the chick blastoderm, which means they're headed toward gastrulation. This upper layer, as we mentioned, is called the epiblast, and there's an area called Kohler's sickle, and that becomes becomes very, very significant. That's viewed here, Kohler's sickle, and it's eventually going to give rise through induction to the primitive streak, which is going to form here. The primitive streak and Henson's node. Henson's node is actually going to become the thing that forms the embryo. So in that sense, it's like the amphibian blastopore lip. And Kohler's sickle is like the amphibian nucoop area, or the fish nucoop area. In other words, it induces the formation of the primary organizer. Well, we'll talk more about that, but just to give you kind of a preview of what this Kohler's sickle does, and it forms here. Here you can see the epiblast. There is a hypoblast layer here, but it's not a continuous layer. It's isolated groups of cells. We can see them here. And they will eventually join together to form the primary hypoblast layer, and then a secondary hypoblast layer will form from posterior marginal zone cells. So they form a layer there, and at this point, from the epiblast of the embryo, you start to get an involution of cells, inward migration to form the primitive streak cells. And the primitive streak cells will give rise to both endoderm and mesodermal cells. So endomesodermal cells, and that happens as development progresses. The primitive streak is here, starting to form, and we'll look at that in a little more detail. And here is Henson's node. It's not labeled, but the significance of Henson's node then is it's like the dorsal lip of the blastopore. So it's the organizer. In this figure, you can see the cell movements in a little more realistic diagrammatic fashion. Here you see cell movements of the primitive streak and the fate map of the chick embryo, and I won't go into the details of the fate map of the chick embryo, but I think it's important for us to understand what the sequence of events are as far as the formation of the primitive streak and the cell movements involved in the primitive streak. Avian, reptilian, and mammalian gastrulation takes place through the primitive streak. If that were a fish, it would take place through the embryonic shield, or if it were an amphibian, it would take place through the dorsal lip of the blastopore. But In this case, it's called the primitive streak. It's in the chicken, the birds, and it is like the dorsal lip of the blastopore. Various kinds of dye marking experiments in time-lapse cinematography indicate that the primitive streak first arises from Kohler's sickle, which we already mentioned. Kohler's sickle was, remember, down around here, and it starts to form and move up in this direction here. Thus, the primitive groove is homologous to the amphibian blastopore. So this primitive groove that's forming is homologous to the blastopore being the opening that will eventually form the archenteron, or the gut. So, although these look different structurally from the amphibian, they are very, very similar in terms of their various cell movements and in terms of what they eventually become. The center of Henson's node will be in this middle region here now. The center of Henson's node is sometimes called the primitive pit, or it's sometimes called the knot. But in any event, it is a little area that is the end point of where there's kind of almost like a zippering effect of the formation of notochord as it comes down through this primitive streak area. And therefore, as this growth takes place, the primitive streak will elongate and it will start to form somites on each side, and you will get the formation of the embryo. You'll get the head folds, the neural folds, and the embryo is now starting to form. Keep in mind, this was the area pellucida on the top of that big yolk. And so it's a small disc on the top of the big yolk, and it's all forming in that little disc, and this is the area opaca on the side. It has some cell structures. They're not going to become part of the embryo proper. They are going to become things like the yolk sac and the allantois. So although they contribute to the embryonic development of the chick, they are not part of the body of the chick, and they do not become incorporated into the body of the chick. Let's now look at the migration of the endodermal and mesodermal cells through the primitive streak. This is now this flat disc that we were talking about. Here we have on the top, this is going to become the epidermal area here. It's the epiblast. Here's where the primitive streak is that the cells can involute through. So what is that comparable to? Of course, in the amphibian, it's the dorsal lip of the blastopore. Here it is, involutes, gives rise to both endoderm cells, indicated in the yellow, and the mesoderm cells. So these cells come down, migrating in from the outside. This is the hypoblast layer, and again, this would be on the inferior aspect of the developing embryo. This shows the involution of labeled cells that are going to go inside of the primitive streak. This is a green fluorescent protein labeled. It looks yellow, but it's green fluorescent protein labeled indicator of where those cells are going in that developing embryo. Here's a scanning electron micrograph showing the area of involution. You can't actually see the opening because there are cells going in there, but notice that these cells have long bottle-shaped structures like this one. These are bottle cells, just like the bottle cells that are present in the amphibian or in the fish, the bottle cells that lead the way of the migration of those cells in to form the mesoderm and the endoderm. If we look at the migration and the molecular mechanisms of migration through the primitive streak, basically there are cell movements that take place, and what directs those cell movements primarily are, well, growth factors, and also there are a whole series, a cascade of those that go through various pathways, including the Wnt pathway that seems to be a major mover in this kind of a migration process. But here you can see Kohler's sickle, which is going to induce the formation of Henson's node, which will give rise to the primitive streak. And Henson's node, of course, is comparable to the dorsal lip of the blastopore that basically is the organizer for the chick. If you look at the distribution of the cells initially, markers have been done, marker experiments which show that these cells will intercalate as this elongation process takes place. It was done a number of years ago by time-lapse cinematography, and they were able to trace the cells and where they went. And these cells then would come up through the middle, and they would come back around, and they would just keep recirculating and at the same time extending the length of the primitive streak. So these cells would be going in this fashion and at the same time elongating the primitive streak and causing the embryo to continue to develop by this intercalation. In other words, the cells more or less taking their turn to get in place to join into making this very important primitive streak that's going to eventually give rise to the developing embryo and to the organism. This is obviously not a diagrammatic representation. These are real photographs of what's happening during chick gastrulation at the 24 to 28 hour period after fertilization. You can see that the embryo is developing. Again, area pellucida, this light area opaca down here, composed of cells that we know now give rise to the extraembryonic membranes. The area pellucida, cells that will give rise to the embryo proper, the embryo body. When we talk about the migration through the streak, we looked at it diagrammatically now. Let's kind of try to look at this in the real organism. The cells will migrate to the primitive streak, just in general, and as they enter the embryo, they separate into two layers. The deep layer that joins the hypoblast along its midline, so they'll be going inside and joining the hypoblast. The second migrating layer spreads to form a loose layer of cells between the endoderm and the epiblast. This middle layer of cells generates the mesodermal portions of the embryo and the mesoderm lining the extraembryonic membranes. The migration of mesodermal cells through the anterior primitive streak and their condensation to form the chordomesoderm also appear to be controlled by certain kinds of growth factors, such as, again, the Wnt signaling cascade, as well as FGF, fibroblast growth factor. But as we look at these structures now and just follow the development, here we see the pharyngeal endoderm. The head process is going to be forming up here. So this is the anterior end of the embryo, the posterior end of the embryo throughout. Henson's node is here. Now you remember this is comparable to the dorsal lip of the blastopore. So as we're moving down here, Henson's node will start to allow the involution of cells. Here's the primitive streak down here. And as development progresses, you start to get the formation of somites. Keep in mind, this is very early in development. This happens before the egg is laid. This is happening very early in that blastodisc. Somites, the neural groove is already forming. The head fold is there. The pre-brain tissue is there. The pre-spinal cord tissue is there. And we continue down through the primitive streak. And here we've reached several somites. Again, the neural tissue is starting to form up here, and we're getting inductive interactions in the embryo proper, this area pellucida, and the extraembryonic membranes as well are beginning to extend out and to form. Some of the mesoderm will actually line the inside of those extraembryonic membranes. Another very interesting aspect of development in the avian system is the axis specification and also the role of gravity in these processes. As a consequence of the sequence in which the head, endomesoderm, and notochord are established, avian and mammalian and reptilian and fish embryos exhibit a distinct anterior to posterior gradient of development, while cells of the posterior portions of the embryo are still part of the primitive streak. The colonizing mesoderm, cells of the anterior end, are already starting to form organs. So let's go back just for a minute to see that. In other words, cells up here are forming organs already, when down here it's just a matter of migration, and not much has happened down here. So there's an anterior to posterior gradient of differentiation. Head first. And then some people have said that's where we get the expression to go head first from the development of these chicken embryos. I'm not sure of the English origin of that, but anyway, it makes sense. So the head develops first, and the tail part of the body will develop secondarily. So part of that is also related then to gravity, at least theoretically. So for the next several days, the anterior end of the embryo then is definitely more advanced in its development than the posterior end, although the formation of the chick body axes is accomplished during gastrulation, and that actually occurs much earlier during really the cleavage stage. So it's predestined during the cleavage stage and actually predestined before the egg is laid. With regard to the role of gravity and the posterior marginal zone, the conversion of the radially symmetrical blastoderm into a bilaterally symmetrical structure appears to be determined by gravity. As the ovum passes through the hen's reproductive tract, it is rotated for about 20 hours in the shell gland, and this spinning occurs at a rate of about 15 revolutions per hour, shifting the yolk such that its lighter components lie beneath one side of the blastoderm. This imbalance tips up one end of the blastoderm, and that end becomes the posterior marginal zone, where the primitive streak formation begins. It's really not known what interactions cause this specific portion of the blastoderm, however, to become the posterior marginal zone, but certainly it appears that gravity does play a role. They also prevent other regions of the margin from forming their own primitive streaks, so that you don't get abnormal embryos formed and you don't get animals that are non-functional. So, the conclusion is, and this is very difficult to get at in a bird system because you're dealing with an egg that's inside of the bird's body, the hen's body, but it does now seem apparent that the posterior marginal zone cells act the equivalent of the amphibian nucleus center. Now you remember what the nucleus center was. The nucleus center in the amphibian, or in the fish for that matter, were those cells that were able to induce the formation of the primary organizer. So they were really the very first inductive cells in the vertebrate system, at least in the amphibian and the fish, and it's thought that that's the same case in chick. And certainly there's significant evidence to suggest that that's the case. So the nucleus center then, in the chick basically, is the posterior marginal zone. And it actually precedes and induces the formation of Henson's node, the primary organizer. So it now seems apparent that the posterior marginal zone contains cells that act as the equivalent of the amphibian nucleus center. When placed in the anterior region of the marginal zone, a graft of the posterior marginal zone is able to induce a primitive streak and Henson's node without contributing cells to either of those structures. So the current evidence suggests then that the entire marginal zone produces various growth factors that will lead to the formation of the primary organizer, which basically is Henson's node, that will lead to the formation of the primitive streak. So let's look now at the formation of Henson's node from Kohler's sickle. Here we have a coronal view diagrammatically of Kohler's sickle area. Here's the epiblast. This would be the head region of the embryo, tail region of the embryo. Here's the yolk material. This is the yolk side, and this is the surface side. Again, you see the epiblast. In this region, you see Kohler's sickle, which is, if we look down from the dorsal aspect, is shaped like this. And at this time then, there are two components, the posterior marginal zone, which we think is like the nucleus region, and here's Kohler's sickle. So we have, these are now labeled, the cells of those components are labeled with different fluorochromes, and you can see that here is Kohler's sickle area up here, going to give rise to Henson's node, and here's an area that represents the posterior marginal zone cells. And so as development progresses, these start to move apart, although this initial relationship has allowed this to become Henson's node. In other words, it's allowed it to become the primary organizer. And as development progresses then, this moves forward. Cells are involuting here and forming an embryo, embryonic body, mesoderm and endoderm is forming as it moves forward. And we can see then that as development progresses, the embryo starts to develop with the inductive interactions from Henson's node that originated from inductive interactions from Kohler's sickle area, which basically is the posterior marginal zone cells. Thus, the organizer of the chick embryo forms from cells initially located anterior to the posterior marginal zone. The epiblast and middle layer cells in the anterior portion of Kohler's sickle become Henson's node. The posterior portions of Kohler's sickle contribute to the posterior portion of the primitive streak. Henson's node has really long been known to be the avian equivalent of the amphibian dorsal blastopore lip, since it is the region where cells become the prechordal plate and chordomesoderm. It is the region whose cells can both induce and pattern a second embryonic axis when transplanted into other locations. And we'll see an example of that in another slide in just a minute. It expresses the same marker genes as Speymann's organizer in amphibians and the embryonic shield of teleost fishes, such as the transcription factor. You may recall goosegoid we talked about as a major transcription factor. Henson's node can also induce neural tissue when grafted into fish, amphibians, or mammalian embryos. It's a ubiquitous kind of inducer. And it's ubiquitous in all of the vertebrates because probably the vertebrates all have common mechanisms. So, as with all vertebrates, the dorsal mesoderm is able to induce the formation of the central nervous system and the ectoderm overlying it. The cells of Henson's node and its derivatives act like the amphibian organizer, and they secrete various kinds of growth factors and various kinds of transcription factors, BMP inhibitors such as cordon, noggin, and nodal, and we had mentioned those earlier just to kind of jog your memory on some of those components that we had talked about in the amphibian. Here is that transplantation experiment that I had mentioned earlier. What you got here is you have a duck embryo and a chick embryo, and it's the same kind of experiment that we've seen previously for the amphibian embryo done by Speymann and Mangold back in 1934 when in their case they took the dorsal lip of the blastopore and transplanted it to another embryo. Well, in this case it's the Henson's node that is being transplanted. It's going from a duck embryo into a chick embryo. It induces a neural tube, and these are some of the derivatives. I won't get into the details of this with the markers, but you get a secondary embryo formed. This duck Henson's node has induced chick material, chick cells, that ordinarily wouldn't form necessarily into an embryonic structure, but it induces those cells to form into a developing embryo. These were cells that were going to form the extraembryonic membranes, and instead now some of those cells chose to form under the direction of the duck Henson's node into a new chick embryo. Hox gene activation is also very important. I won't go into the details of this other than to mention that when we talk about anterior-posterior patterning, we talked about the head first sort of thing. What that entails is Hox gene activation. With regard to anterior-posterior patterning, the patterning of the definitive anterior-posterior axis occurs differently for the mesoderm and the neural ectoderm. But in both cases, the process involves timing, in other words, the sequential generation of cells from a zone of undifferentiated proliferating cells and the influence of caudalizing molecules. Therefore, the anterior-posterior identities along the axis of the hindbrain to the tail are specified as a function of the time emergence from the primitive streak and Henson's node, the head-first concept we talked about earlier. It has been proposed that the length of time cells are resident in the primitive streak region determines which Hox genes are expressed, and this pattern of Hox gene expression can also be under the influence of FGF retinoic acid gradients. Just as in Drosophila embryos that we talked about earlier, the Hox genes specify the identity of the cells along the anterior-posterior axis, and the lower number cells are basically associated with the anterior region, and the higher number Hox gene cells are with the posterior region. So the take-home message here is that the Hox genes influence the anterior-posterior patterning. Anterior Hox genes, which are identified with lower numbers, are expressed early and extend farther anterior than genes such as HOXB9, which is expressed later. This does not extend as far anterior as the lower numbered Hox genes, but only in the posterior component. We also have a left-right asymmetry in the chick embryo. The vertebrate body certainly has left-right sides. We've talked about that before. The heart and spleen, for example, are on the left side. There's also a model that's been expressed for generating left-right asymmetry in the chick embryo that relates to various kinds of cascading genes. I won't get into the details of these genes, but basically left versus right has a different set of growth factors and genes that influence left-sidedness and right-sidedness in the developing embryo, and different genes are expressed that make left side structures and right side structures. The vertebrate body, certainly we know, has distinct right and left sides. The heart and spleen, for instance, are generally on the left side of the body. The liver is usually on the right side. The distinction between the sides is primarily regulated by the left-sided expression of actually two proteins, it's thought, the paracrine factor nodal and the transcription factor PIT-X2. But we won't get into the details of that, other than to say that there are certain growth factors and transcription factors that cause left and right-sidedness in a developing embryo, and if that doesn't happen, the embryo will not develop normally and will probably not be viable. So as we conclude chick development, we've learned that it is very, very similar in many ways, at least the important components like primary induction and induction of certain tissues, virtually identical to what we've learned about in the amphibian and fish. The same is true with respect to axis gradients, anterior, posterior, left, right, and so forth. The other thing that I would mention in concluding is that there are a variety of complex gene activities and transcription factors involved in these various kinds of activities in the chick. And as we learn more and more about the chicken, we learn more and more how similar this is to all of the other vertebrates that we are learning about.